This is episode 50 of the Immunology Podcast, IUIS 2023, where immunologists meet with Drs. Miriam Merad and Mark Davis. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Drs. Miriam Merad and Mark Davis from IUIS on the podcast to talk about what to expect at the 2023 IUIS Congress taking place in Cape Town, South Africa from November 27th to December 2nd. We've also got our usual roundup of recent highlights of immunological news coming up. But first,
talking about conferences, Immunology 2023, which is the annual meeting of the American Association of Immunologists, will take place from May 11th to May 15th in Washington, D.C., right there in America. For most up-to-date information about Immunology 2023, including the scientific program, very important, you can visit www.immunology2023.org.
All right, we have a bunch of trips coming up, Brenda.
We do. The podcast is going on the road. Are you excited?
I am. So, we got the one in May coming up, and then we're going to South Africa right at the turn of the month, which is going to be exciting. I've never been there. I don't know if you have.
No, I've never been anywhere in Africa, so super exciting.
Another continent off the list. Same here.
Yes. Yeah, bucket list thing. No, I've never been. I do come from the Southern Hemisphere, so that will feel familiar enough for me. So have you been south? In the Southern Hemisphere?
No, I've been equatorial, but that's about it.
Oh, well, you are in for a treat. Everybody knows it's the best hemisphere.
Well, it's going to be summer when it's winter up north, so that'll be interesting.
That's true. Hopefully the weather will be nice. End of November, beginning of December.
Early summer, right? Like if I do this right?
It's like late spring, right? Summer would technically start in December 21st.
Okay. So this will be like May. So we're going to two conferences with the same weather at opposite times of the year. There you go. 6 months apart. Hey.
Yeah.
Like we can wear the same outfits. We can like match it all up the same way.
We can wear like colors, but then upside down or something like that.
That's going to require a black shirt and purple shorts or pants. That's going to be weird.
No, let's not do that. Let's not do that. But talking about upside down, you know that the stars in the Southern Hemisphere, the few constellations that we can see from both sides, they look upside down in the south. So I always remember, Orion's belt. We have a different name. Like, I don't know if it's where it comes from. We call it the Three Marys, probably some Catholic thing. So you have the Three Marys, which are the Orion belt. And then it's always funny because people say, yeah, that's the belt, the Orion belt. And then it has like a little dagger hanging from the belt. And then when you see it in the Southern Hemisphere, the dagger is upwards, right? It's like, yeah, the guy is upside down. Makes no sense. And then when it came to the Northern Hemisphere, all of a sudden, the guy is up straight.
I think looking at the constellations and saying, I can see a dipper. I get that one, right? That's the easiest. But a lot of the other stuff, like, yeah, can't you see how it combines this way? I'm like, you were on some shrooms when you put those together and decided it was a shape. I'm sorry.
Well, you have the Southern Cross. That's very nice. It's a very nice concept. Very easy to find. It's cross-shaped. It has like 5 little stars. And then he tells you which way the South Pole is.
Because of how we spin, right? You can do latitude by the stars, but you can't do longitude by the stars. That's the whole problem, which is why you have to have star charts, because the stars are fixed latitude-wise if you go up and down, but longitude, they change. For navigation without fancy things, you have to have star charts for where they are at a certain time of year and the longitude you're at, because it all changes.
Yeah, or better, have very precise watches. And then that's how the Brits did it. The whole story of the precise, you should go to Greenwich, to the observatory in London. They're very nice. But we are digressing. We're not an astronomy podcast, are we? Although it's a very nice discipline.
Keeping things engaging here, I'm going to talk about how tumor PD-L1 engages myeloid PD-1 to suppress type 1 interferon to impair cytotoxic T lymphocyte recruitment. Published March 13th, Cancer Cell. Last author is Kaibin Liu. First author is John Clement. I also think the second co-first author is Priscilla Red, based on the footnotes. There we go. Yes, correct. All right. So, PD-1 blockade, it's all in vogue. PD-1 ligand with PD-1, makes cells not want to kill bad things, right? Causes immune suppression. High level story, right? We all know it makes fancy new immunotherapy drugs blockade this pathway. Something that they found, though, is that it's not very consistent across tumor types, primary versus metastases, and other engagements. And there seems to be effects going on that are not just related to suppressing cytotoxic T cells. So we know that the PD-1 pathway can suppress cytotoxic T cells and prevent killing. But figuring out how else it's working has been difficult to a certain extent. So this paper looks at co-cultures, mostly of tumor cells and tumor-specific cytotoxic lymphocytes. And they do this combination of mouse systems and ex vivo and just cell culture, and then validate some of this in humans at the end, like one must do. But what they demonstrate is that obviously if you delete the T cell PD-L1, you have decreased lung metastasis in a CTL-dependent manner in the mice. So the cytotoxic PD-L1, the ligand part of the thing, they're both actually receptors. They're transmembrane proteins. So, but at one's L1, one is not. It has a role in affecting lung metastasis, but not necessarily other models of tumor. But it's not the direct actor in this process. What they find in this paper, and they do some tracing and then all the requisite knockouts. And so how they kind of get to this whole conclusion is through some combination of knockouts, looking at RNA-seq for signaling and seeing patterns of other immune cells that are really important for the response they see that weren't the cytotoxic lymphocytes, and then going back and doing more knockouts and validating it. And what they find is it's actually the myeloid cells, specifically the macrophages, that have the PD-1 on it, that the cytotoxic lymphocytes are interacting with. So what's happening is that the T cell PD-L1 engages the myeloid PD-1, and that activates SHP2 signaling, which is a known part of the PD-1 cascade, to antagonize the interferon-1 and STAT1 pathways and impairs cytotoxic T cell recruitment to lung metastases. So it's actually the myeloid cells who have the PD-1 going on. And so PD-1 blockade in some of these tumors is dependent on there being a myeloid working, depends on there being a myeloid cell population in the tumor, right? So kind of the hypothesis then in human disease that's responsive to PD-1, you have a good amount of myeloid cells sitting in those metastases. And so they're being signaled by the PD-1, right, to not do something. And so when you remove that, then you have T cell engagement. You then have that pathway blocked. And so now the myeloid cells are going to drive cytotoxic T cell recruitment. But it's not just PD-1 on T cells doing this. It's the PD-1 on the myeloid cells interacting with T cell PD-L1 at baseline in this immunosuppressive tumor promoting environment that allows the tumor to progress, the metastases that are occurring. And so if you blockade that on the myeloid cell side of PD-1, then you can get re-engagement of the pathway. So it's giving us a key insight here in this paper about where the other cell actors are that are driving this.
Okay. That's very interesting, like looking into PD-1 on myeloid cells, because usually people always think of even like cytotoxic T cells, like most of the focus has been on what PD-1 is doing on those cells. So I find it very interesting that it is, in this case, they find that is the myeloid cells that are actually suffering more. And also I like it because often people focus a lot on the effect of PD-1 signaling on like TCR signaling, when actually also PD-1 is also involved in other things, also within the T cells. So, And so what I didn't get too much is the difference between primary tumor and metastases in a sense.
Yeah. So this is, they're finding that this TPDL1, the T cell.
The tumor. No, I think it's tumor PDL1.
Yeah, it's tumor. Sorry, PDL1 enhances. Mm-hmm. It's not, regardless of how big the primary tumor grows. Mm-hmm. This is what's engaging for the metastases, right? So it's not necessarily the same pathway, is what they get at. So they look at the tumor growth site versus metastasis, and are finding out that knocking out the pathway doesn't necessarily have anything to do with the primary tumor, but has to do with the downstream metastasis control. Because the tumor engagement is signaling the macrophages to shut up and not recruit the cytotoxic T lymphocytes. Mm-hmm. But that's really about metastatic control, right? You're going to have the tumor growing and setting stuff off. Once it starts seeding, the macrophages go, what the hell is this? And kill it, unless that tumor is signaling the macrophages to shut up.
Okay. Very interesting. Oh, always other immune cells getting in the way of the work of the T cells.
Macrophages, we've discussed, are very important.
Yeah, but they're double-faced. You have good macrophages and you have bad macrophages.
So that's true of all T cells too.
No, you have good T cells and confused T cells that want to be good, but they're just not. And macrophages, they're just.
What is? Oh, come on! A T cell against yourself is not a good T cell. It's not confused. It's turned evil. It's gone to the dark side.
Okay, I think you're right. Oh, all right. Very interesting. Thank you for sharing this new perspective on PD1, PDL1 interactions, and I'm going to talk about a paper. Actually, this is a publication from our guest today, Mark Davis. This is actually the pre-proof, so it is fresh out of the scientific oven. But I thought it was really nice. I saw some comments online, and I thought it was a cool paper to just mention. Robust T cell responses to Pfizer-BioNTech vaccine compared to infection and evidence of attenuated peripheral CD8 T cell responses due to COVID-19 is the title of the paper. It's going to be published in Immunity. Was just accepted about a week ago. And first authors, Fei Gao, and as I mentioned, from the lab of Mark Davis. And basically in this publication, what they look, they take a kind of close look into into T cell, spike specific, and also non-spike, other kind of SARS-CoV-2 specific T cell responses. They focus mostly on spike specific. And by using a new platform for staining, they call them spheromers, which is kind of a little structure made of ferritin, of a kind of a ferritin-derived structure that has on its surface 12 peptide MHC molecules that allows for a kind of a stronger stain and binding to the peptide-specific T cells. So they use this for both CD8 and CD4, so MHC1 and MHC2 restricted TCRs, and peptides, and they use this to look into responses in patients and in people vaccinated. And they do kind of a really deep dive into the kinetics of the T cell responses upon either vaccination or infection with COVID with the original. So their samples were taken in kind of the early COVID times. And they also see what happens when people that had COVID are vaccinated on top. And I thought it was very interesting because so I think that the whole idea is quite straightforward. They basically have these pheromers. They have samples from various patients, and they look into binding to peptide MHCs against dominant kind of. They choose a couple of MHC molecules. I guess they choose the ones that are most common amongst their patients, and they pick up certain peptides that are predicted to be presented on these MHCs, and they use those. So they have several different peptide MHC combinations, and they have two different ones for MHC1 and one for MHC2. And they look into the serum, the plasma of the patients. And basically what they see is that what I think the highlights of they do a lot of analysis, but I think the highlights on kind of the big picture is the fact that when they look into the upregulate, kind of the increase in cell numbers of CD8s and CD4s upon vaccination first, they're intrigued by the fact that they see that there's a difference in the kinetics of kind of T cell expansion between CD8 and CD4 cells. And they see that, and they suggest that this is something that might be specific to mRNA vaccines, because they cite other conditions in which this doesn't really happen. And they see that whereas the T cell, the CD4 response is kind of peak quite early on, about a week. I think week or 10 days, you have already kind of the peak amount of tetramer positive T cells in the CD4 compartment. The CD8 compartment keeps expanding and keeps inflating up to 40 days after the end of the vaccination regime. And so they suggest they see that this is different, and they're not sure why this is, but they see this specifically for the naive vaccinated individuals. And then what I think is really cool is that they have some patients in which they had symptoms of COVID. I think at that point it was not that easy to get actually tested by a PCR, things like that. It was early COVID, but so these people are kind of confirmed COVID patients, and then they get so they also look into the growth of the CD4 and CD8 populations, spike specific, and they see lower amounts of tetramer positive cells, or like spike specific cells. So in the case of the vaccination, you get after the second doses, you can get almost 20% of all the CD8 cells in these patients are spike specific, but this number is much lower for people that had actual just an infection, a COVID infection. And also what is interesting is that when they look at the immunodominant peptides that are characterizing this, because they test different peptides, right? And they see that there's a different selection of immunodominant peptides between people that were vaccinated and people that had a natural COVID infection. So it's also very interesting to see that there's a fundamental difference in how the immune system is seeing the virus, and is seeing the spike protein either in from the virus or from the vaccine. But I think what caused a little bit of kind of talk in the neighborhood is the fact that they see that in the case of people that are vaccinated after having had COVID, they seem to have a lower antigen specific CD8 response. So they have a lower frequency of tetramer positive cells after vaccination than people that were vaccinated while being naive against COVID. So suggesting that there's some kind of reduction in CD8 T cell quality, or something about the T cells, that the CD8 T cells that gets affected by a prior COVID infection. And this is not, they don't see the same for CD4 cells. So CD4 cell numbers are similar between naive and previously infected patients. So this is something specific about CD8. And the authors do mention, and we do know that COVID infection seems to affect MHC1 expression. So COVID does have some immune evasion techniques, which might explain the reduced CD8 positive cells, so COVID positive, COVID specific cells in these patients. But it's unclear. But I think that in general, I thought it was very interesting that they see this. And I guess there's a lot of debate nowadays. How much does COVID affect, negatively your immune system? Is this worrisome? Should we be worried? Is it generally applicable? Is something that you see in many types of infection? Because we also know that other infections also affect your immune system in a way. So, but this seems to be something very specific about the CD8 T cells. So that's basically, I think, the highlights of this paper are about these results, and how they look at the very they could look with using these spheroms very closely into both CD4 and CD8 responses and their kinetics.
Very interesting. I mean, I think the final point you made as well, like, what does it mean? It's also interesting because it's basically ubiquitous a virus now. We can't avoid it, right? You're going to get it. You're going to get it again. Then you're going to get it again years later. So what does that really mean? And are we measuring things that happens in other viruses all the time that we never bothered measuring before?
Yeah.
How much of this is really specific to this entity? Are we just uncovering biology? And heck, maybe the hygiene hypothesis of like more autoimmunity comes from the fact that if we're not as exposed to many viruses, we don't have enough tamp down of our immune system by all these infections we get. And so we have too hyperactive immune system. I mean, who knows? Right? Maybe this is normal, and actually, why no one wants any given disease, getting some infections is what's required to have you not have crazy, Darth T cells, to call that in our previous discussion.
Yeah, I mean, in the end, our immune systems evolved under constant pressure, under constantly infectious pressure. So I guess that I don't know if it's desirable to have infections, but it does seem like our immune system was developed in an environment, it got used to being perpetually challenged, and the fact that we don't do it might we might not be ready. We might not, our biology might not be tuned to our very hygienic present.
Sequence everything. Yeah, exactly. We know it all. Like, maybe we don't want to know about maybe it's disturbing to know all these things, right? Oh, it is suppressing you, but actually it's good to suppress you. All right.
What you got?
I don't have a segue to this one, but I'm a big fan of the lab it comes from. So this is in immunity. It's endoplasmic reticulum stress in the intestinal epithelium, initiates purine metabolite synthesis and promotes Th17 cell differentiation in the gut. It's from the Blumberg lab at Brigham and Women's / Harvard. First author is Jinzi Duan. So this relates to my field of work in IBD, actually. We know that disrupting the unfolded protein response and disrupting XBOX1 binding protein can lead to IBD manifestations. That's some of Rich's longstanding work. He's kind of known for that, as well as developing the FC receptor for biologics, or his two big things. Or at least being a major contributor for that. So when we think, there's two first authors. The other first author is Juan de Matute. Caught that in a footnote. And so we know that microbes generate a push towards IECs, towards microbial adherence to intestinal epithelial cells, generates a push towards Th17 cell differentiation. But how this happens, no one knows. What happens is if you have bacteria that are known to do this, and they map this out, notobiotic experiments, knockouts, everything, that these bacteria that do this generate unfolded protein response in IECs. That leads to a downstream signaling cascades that involve both reactive oxygen species and purine metabolites. And that reactive oxygen species and purine metabolites, because if you go to that step of the pathway and blockade those with antimetabolites, allopurinol and acetylcysteine, those downstream readouts of the unfolded protein response cause the generation of Th17 cells. So they have like an inducible model that they do here for unfolded protein response, which is pretty sophisticated. So they have the conditional knockout, but they also they have a drug-inducible system for inducing unfolded protein response. And they put that into germ-free mice and show that you don't need the bacteria, you just need the unfolded protein response. They also do this with just the bladed knocking out XBOX1 binding protein, which will cause an unfolded protein response. And so they use a couple of different systems to lead to showing that the bacteria cause unfolded protein response. Unfolded protein response causes upregulations and RUOX and everything else. And then it comes back, and what ends up happening is that oxygen sensitivity, or then the immune cells will sense that oxygen tension change in the purine metabolites, and that generates the whole Th17 process. So I thought it was cool because it A lot of microbiome papers like, the microbiome causes this or that. But this actually gets the signaling that the bugs do that can be recapitulated without the bugs. And then the signaling without the bugs is demonstrated, and then that leads to the downstream pathways. So, they know that this is associated with IBD in various ways in terms of high levels of Th17. This genes that are involved here are involved in IBD with various SNPs. But the point being, it's really demonstrating a pretty good pathway to show how a signaling pathway induced by bacteria leads to downstream immune differentiation. They map it out. So I liked it.
So again, the bacteria telling us what to do. Is that it?
I mean, we're just, giant meat necks for bacteria.
Okay. So I see why you like the paper. It's interesting. It's an interesting approach. All right. Wrapping up today our lineup of papers. I want to talk about another paper that caught my attention, because also when I read the title, I thought, whoa, what about this? And then I have to say that I was a little bit underwhelmed, but I guess it's still pretty good. So this paper is called "The Dietary Sweetener Sucralose is a Negative Modulator of T-cell Mediated Responses." So my question to you, Jason, is: do you put sucralose in your coffee?
No, I use real sugar or nothing at all. But mostly, I do not have sugar in my coffee because I like my coffee dark like my soul.
That makes sense. That checks out. But yeah, so a lot of people freaked out, I think, about this. It's like, oh my God, our artificial sweetener is killing us. I mean, I'm gonna kind of spoil it here, they're not, but it's interesting. So this paper was published in Nature. First authors Fabio Zani and Juliana Blaghi, from the lab of Karen Vousden at the Francis Crick Institute in London. And basically, they started with the idea. So people seem to there was some idea about, like, are these sweeteners affecting our immune system or not? So they decided to kind of look at it head on. So we know that sucralose is a calorie-free sweetener, so it doesn't affect the metabolism in principle of cells. It's not metabolized. But we do know that we have some limited absorption happens when you consume it, and that if you consume 250 milligrams of sucralose, you can have plasma levels of about 1 micromolar after about an hour and a half. So they looked into the maximum, the recommended maximum intake through, like, the regulatory organs, either the FDA or the European equivalent of the FDA. And they used this to kind of interpolate the plasma concentrations and try to interpolate how much sucralose that would mean in a mouse. And so they basically came up with this amount of sucralose that would replicate this maximum intake in humans, but in mice. And then they tested what happens when you do the T cells and to the immune system when you feed the mice this amount of sucralose. And again, long story short, they look into a bunch of things. They don't see major effects on myeloid populations or on B cells. But what they do seem to find is that T cells seem to be affected by the presence of sucralose, so by the consumption of sucralose by mice. So one of the examples that they look into is transferring T cells into RAC2 mice, which then would proliferate. This is normal homeostatic proliferation that happens in these conditions, and they do see, like, there's a reduction in the proliferation of donor T cells in mice that are consuming sucralose. They see reductions in proliferation in vitro, lower polarization into T helper subsets, and they look a lot into what's going on. They look into particular receptors for molecules like sucralose, and they end up kind of homing down on that this is actually something that's TCR dependent, because they see that if you induce proliferation of T cells by high amounts of IL-2, for example, this inhibition is bypassed. And also if you activate the cells with PMA and unamycin, which of course bypasses completely the TCR, there's no effect of sucralose presence in the cells. So they look into different, so they kind of realize there's somewhere downstream of the TCR that this is happening, somewhere around the TCR, but not too much lower, because PMA unamycin would show that. And they end up showing that there's a delay in the phosphorylation of one particular protein. This is phospholipase C gamma 1, which is, of course, was discussed in our last episode, talking about the TCR signaling. So this phospholipase goes downstream of TCR, SAP70, LAT, and then kind of is part of this cluster that forms around the TCR. And the function that it has is to cleave phosphatidylinosytol biphosphate and release deacylglycerol and inositol 3-phosphate, which then continue downstream signaling, including ERK activation, and importantly, calcium release from the endoplasmic reticulum stored. So what they show is that basically you have a kind of an interruption of this part. So there's problems with calcium signaling and the efflux of calcium upon TCR engagement. They don't really know what exactly is going on. They don't give us a particular mechanism on how sucralose is interfering with the function of the phospholipase. They do some kind of interesting. It's an interesting protocol which they use a machine called Orbi-Sims, in which they have like a cryo-EM mass spectrometry. I did not completely understand, but basically they can look. It can do imaging of chemicals on the surface of the cells with like micron resolution, and they can detect which are the particular chemicals. And that's how they see that sucralose is not really getting into the cells. They don't see it going through the membrane, but it seems to be interfering with the proper composition of the membrane away, and that prevents the phospholipase from kind of getting in the right place at the right time and being and acting downstream. So I thought it was a little bit sad. It was not completely clear what's going on, but that's as far as they get. And they do show that there's some functional consequences of this. It's to do some reduction. So at this what they consider rather high doses of sucralose, they see reduction in some T cell function, reduction in tumor control. But what they focus on, and they're kind of trying to see the glass half full, is that they can also help reduce the severity of some autoimmune models. They have some diabetes models in these mice. And it shows that they can actually reduce the onset of diabetes in these mice. And so basically the bottom line of this paper is that no, sucralose, the amounts of that a normal person consumes probably is not doing anything. These amounts are very high. But they like to suggest that maybe, given the sucralose is safe otherwise, could be used as a treatment for autoimmunity or for reducing, actually for dampening T cell activation in patients. So interesting. I think was like, it felt very controversial at the beginning. In the end, it's like, man, you're probably fine eating sucralose. But maybe if we pump you up with a little bit of that, it could help you, could be a alternative immune controlling drug. I don't know what you think.
It's interesting. I don't know how much you can pump it in. You'd have to like IV it or something, because if you have too much of this, it kind of gives you GI distress. I was going to ask what the microbiome was showing.
So they don't see, they looked into, I forgot to mention, they do like, they don't see changes in the microbiome that would explain the results.
So this effect doesn't happen in germ-free mice, that they actually go that far?
I don't think so, but they do see effects on the proliferation in vitro.
Okay. I was wondering if like sucralose is being metabolized at all, and that's what's causing some of this, is a metabolite of the bugs of sucralose, because they can chew it up and we can't.
They don't see, no, they just see, we find no consistent shift in bacterial species, but they don't do it in germ-free mice. It's true.
Well, We're going to be having a great conversation here soon with Doctors Miriam Merad and Mark Davis about a conference. But if you are going to a conference soon, like Immunology 2023 or even IUIS Congress, or if you're looking just to expand your network, make the most of your experience by downloading our collection of tools to help you prepare for your next event. STEMCELL Technologies downloadable checklists and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the conference toolkit at www.stemcell.com/conference-toolkit.
So today we have a very special episode. This is also as we mentioned, our 50th episode with the podcast. And we're really happy that we're going to discuss a very nice upcoming conference with two very special guests that are actually coming here for the second time. So we have with us Professor Miriam Merad. She's from the Icahn School of Medicine at Mount Sinai at New York, and she has already joined us to talk about her research last December. And we also talking to Professor Mark Davis from Stanford University, and he is also going to talk with us about the conference from the International Union of Immunological Societies that is going to take place this year in Cape Town at the end of November. So thank you so much for joining, and we're very happy to have you both back.
My pleasure.
Very excited to have you here. All right, so I'll start first. There's international conferences and then there's international conferences. What I mean by that is a lot of international conferences are in Western Europe, and this is not that. And so I think just diving in, you're hosting a conference of immunologists in South Africa. What does that look like from a conference planning and an objectives that really makes it stand apart and unique from kind of what I think most people think about who probably listen to this podcast in terms of conferences?
Maybe I'll start just because I was part of the early lobbying to have this conference in Cape Town. Yeah, I mean, the international conferences have typically been in developed countries in Europe, Japan, more recently China. Areas that had a lot of infrastructure, a lot of money in terms of research. But we made international congresses, which have been going on since I believe this 1960s. In fact, this is the 40-year anniversary of my first international congress, which was in Kyoto, Japan. I was just a child at the time, but it made a big impression on me. We made the argument, Clive Gray and others in South Africa made a successful argument that we'd never had the International Congress, even though immunology is very international. Lots of places are doing it. I mean, it's a huge, the immune system is a huge life support system that's really important to basically everyone, and especially in areas where you have these terrible diseases. I once was trying to persuade the Gates Foundation to work on flu, and they said, well, no, Mark, the people we care about, flu is the least of their infectious disease problems. They've got much more serious stuff going on, and that's what we're focused on, and that's totally appropriate. But so anyway, we actually were pre-pandemic saying, well, immunology has been largely focused on first world diseases like autoimmunity, and not enough focused on third world problems like infectious diseases like malaria, like HIV, like TB, and that was our argument, that we should not only will it help the people in those countries to have this international activity, and immunology has been such a happening field, but it would introduce international immunologists from the first world to the problems of the third world, and that was what we proposed, why we said this is a unique opportunity. We should be doing this here. And then the pandemic hit, and it delayed by a year the conference. It should have been 2022. It was actually, now it's 2023. But if anything, the pandemic has made it clear that infectious disease is really a thing that isn't just in third world countries. It isn't just a problem there, but you might have noticed it was kind of a big problem everywhere, and we all had to accommodate it, and many people died or have lingering health problems from this really big pandemic. So that helped. That I think that helped seal the argument. I mean, the International Committee had already decided on Cape Town and for those reasons, but if anything, the pandemic has made it even more dramatic how we need to think more about infectious diseases and vaccines and things like that. Vaccines have not been popular in academics. They were considered to be, okay, Pasteur figured this all out, and now it's just up to the companies to do what they need to do. And then, but the actual fact is the companies have not put much into the research into vaccines. They're still doing what Pasteur did, even when it doesn't work. And the diseases like HIV and TB and malaria are, another dimension of difficulty compared to the ones that have worked based on what Pasteur did or some of the modern variations. So an important thing is also to re-engage the academic community in trying to understand vaccines at the level that we need for HIV, malaria, and TB.
I so much agree with you that there is so much knowledge that happened in the last decades, and yet there are diseases. I am from North Africa, from Algeria. There are diseases that are still occurring, prevalent, that would benefit tremendously from a lot of development in the Western world. And I think the goal really, first, it's a statement that all of us are going there, but also that we need to mobilize knowledge to really continue to understand how we can cure, prevent very prevalent disease, and this idea that there are some disease in the low and middle income country and not in Western countries, so we shouldn't care about this. I think COVID prove us wrong, right? We are super global now species, right? Because we travel and microbe can evolve. It's very important that we think globally about everything that we do. And goal here is to really try to think about problems that are endemic in Africa. And I think by going there, and I have to say the international community have been extremely enthusiastic about going there and meeting African scientists. I think this is what we want to achieve, is really making sure that there are problems there that needs to be addressed full force, and bringing the recent immunology development and knowledge that is really tremendous to these problems.
One thing we hope to come out of that are collaborations that with more awareness of immunologists in the first world of these problems, they could be forming productive collaborations with people on the ground there, and I think that would be one of the best outcomes.
Yeah, it's so true. So there's an example of I'm working on a disease not many people have heard of it, called histiocytosis, and the only reason why I worked on it is because a previous colleague, phenomenal immunologist Ralph Steinman, who had the Nobel Prize for the discovery of dendritic cells, once organized a meeting on this disease in a place where we saw patients suffering from diseases. We all went there and we decided, well, we're going to spend a little bit of time working on the disease. And many discoveries came out of this conference. So things, tremendous things can happen when, you bring people together and you highlight, what are the problems that we need to really think about. So the hope is that lot of collaboration, indeed, as Mark pointed out, will really be initiated there and then continue to develop after the meeting.
Yeah, so I was going to say that I see a lot of interesting themes. What you mentioned, so the idea of bringing global immunology to third world countries, or like to somewhere outside of the main focus of research, because to bring this expertise to the ground over there, to expose people that maybe are always done their research in very kind of Western problems or diseases that are characteristics of very highly industrialized societies, and then they forget that, as you mentioned, all these things, they still exist. They just don't see them in their everyday life. I think that's super interesting. Also, for me, coming from the global South as well, it is good sometimes to have these kind of effort to bring the latest and the community to this area, to the global South. And just want to say that just for our listeners, because maybe not everybody is aware of the International Union of Immunological Societies and kind of the ethos of this association and what it's composed of. So would you like to maybe just give me a very quick comment on what the IUIS is?
Yes, it was created in the 60s, in fact, by a group of immunologists from the Western world, Europe and America and United States and Canada, to really think globally about immunology, right? So the idea is that all the immunology society from specific countries or regions will get together regularly to first organize this type of big conferences and making sure that everyone gather in one places, but also organize committees that to focus on big problem in immunology. So there is a committee that thinks about clinical immunology, immune diagnostics, veterinary immunology, vaccine committees, and these people are usually the members of these committees, are also sitting in other societies in their own country, but they are thinking more globally about specific problem. And so I became the president in January, and so I'll be president for 3 years. And really the goal is to enhance collaboration between countries, think about global problems. So, for example, there is this, when we are thinking about immune diagnostics, there are very few immune diagnostic tests to really measure how we are doing immunologically. In fact, I would like Mark to comment on that, because Mark has been saying this for a long time, and he built a whole infrastructure to really measure the human immune system. Yet, we want to bring this to the clinic so that, in our routine checkup, we could at least measure how we are doing immunologically. And this is a complicated process that requires with different type of regulation in different countries. So we think about this, in globally. There is a big focus on vaccine and vaccine distribution. So we can make phenomenal vaccine in one place, but how can we get it, to be distributed in this? So during COVID, we thought a lot about this. There is also knowledge, how we can bring different knowledge to the less exposed, right? So this is the goal of this International Union of Immunology Societies, which should be the mother of all societies. And in fact, all our members are, as I've said, belong to other society. And the big mission is that big congress. And the big congress, this is where there is a larger scientific, it's a beautiful congress, really, because there's a larger scientific committee from people from all over the world, and we think of what to highlight here. Mark is leading the effort, he's the chair of the scientific committee, and he insisted on, focusing on infection disease and focusing on all the immune response to infection and vaccines.
How often is the Congress taking place?
Every 3 years. And it's a very serious endeavor because we vote for what country is going to host the next one. The next one will be in Vienna in 2025. Now it's 2 years because we delayed by 1 year, this one. We're hoping that, it will continue. It should continue to circle across continent.
You have a conference every 3 years. It's international focus. What are your main themes for this specific conference? Our listeners are listening to the podcast. They're going to go, well, they're thinking about going to this. What are they going to get here specifically this time around?
We're trying to cover the waterfront. We clearly have emphasized infectious diseases and particularly highlighting with special programs the serious diseases of the African continent, malaria, HIV, TB, but we don't want to miss. It's a big tent. We want everybody to have some piece of the action here, and so there's cancer immunotherapy is a huge deal clinically. It's transformed oncology treatment and promises to be even better. Right now, a fraction of people are being cured, which is a miracle. Which is wasn't happening before with chemotherapy and radiation, but now with smart immunology, a significant number of people are actually being cured or have remissions for that last quite a long time. But there's still a lot of room. There's still a lot of people that are not benefiting enough, and so it's a huge area of research, a huge area of commercial research, and we have industry sponsors and participation. We have speakers from Moderna and I think BioNTech also to talk about vaccines and about they've been leading the kind of most modern immunology effort in the vaccine world. Immunology touches almost every disease. And I tell the cardiologists at Stanford that cardiology is a sub-discipline of immunology. They don't take that very well, but there's definitely there's an intersection there that is going to be really important. Is building and is really important, and we want to tell people about that. Computation is also huge in immunology. There was this mantra for many years about hypothesis testing and mechanistic experiments being the gold standard, and that works reasonably well with experiments with mice, but it doesn't work well with experiments with humans. And that doesn't mean humans aren't important or that we don't need to get that data, but it's a different kind of data. We have to think of it differently. I tell people it's ignorance-based science that we don't know anything, so let's just go in there and start measuring stuff and see what falls out, what giving us a clue, and then you can deeper into that disease or into that phenomena. So anyway, immunology has been an incredibly dynamic field. Or like every week I've been in it, and it's just it's constantly evolving. It has a lot more room to go. We need to make better vaccines. We need new ways to treat cancer, building on what has been successful. We need new treatments for autoimmunity. All the treatments we have for autoimmunity right now are palliative, that they reduce the frequency of autoimmune incidents, but they don't cure. Nothing cures the disease, and the reason is we don't know enough about it. We don't know really enough about why it starts and then why it doesn't resolve. It seems to go into its own thing that is perpetuating and, of course, debilitating the people. So there's a lot of medical mysteries that where immunology is really a strong part of it. So and that's what makes it exciting from a research point of view, is that not only are there new things to discover, especially in humans and human diseases, but if you discover something, you can almost immediately think of how to apply it to patient care and to some of these diseases. And there's a whole pipeline of basic scientists, physician scientists, industry scientists, and productions and development of medicines and so forth, which is, I think this is the most, I think I'm somewhat biased, but I think it's the most happening part of biology today.
We will have a lot of neuroimmunology session, aging, dementia. Yeah, there's a lot of data that inflammations are really driving the aging process, or at least contributing. So we will cover all these aspects. But there's also another aspect of this meeting that's as important, where we will have roundtable discussion or debates on big issues, in science and society. So we will be talking, for example, about data sharing. So how do we share data efficiently? We will be talking about knowledge, like how do you really educate the public about, what we are doing. We saw the lack of knowledge, which was very helpful during the pandemic, right? The public was super confused by the scientists, and some non-immunologists started to say, well, in America, there was not only non-immunologists that hurt us, but in any case, I think education is going to be a big part of this of the meeting. How you educate, how you fight misinformation. That really is an important subject for us. It's a complex problem, but how can we build tools to fight misinformation? We will be talking about the future of publishing. Publishing science. It's a big problem of how that maybe is technical here. I don't have to go into details, but there is a lot of open access journals and how what the future looks like. We will be talking about building capacity in low and middle income countries, and we will have lots of very exciting debates and roundtable discussion on these. We will be talking about about vaccine distribution, which I mentioned earlier, and how people in low and middle income countries face the COVID pandemics and some of the resources they built, and how some of them were penalized. So there is a whole set of also discussion that's going to also be happening in addition to the dive into our scientific field.
So I see there's a lot of room for people that attend the meeting, not only to go for the science, but also for the role of immunology in the larger picture, kind of in and guiding a lot of the things that actually affect people's lives, even outside of the academic. I think that's very cool, because it's important to give space for those conversations. And I also want to say that maybe moving a little bit away, I'm going back to what Mark was mentioning, this idea of having people from different areas and kind of showing how there's a connection between the academic research, the industry, and how we get those advances into the clinical care. And I just wanted to point out for our listeners, because I thought it was really nice, that there's a couple of really interesting, like, for example, keynote lectures from very prominent scientists, Nobel laureates among them. So people like Emmanuelle Charpentier, James Allison. So Emmanuelle Charpentier, of course, Nobel Prize for her co-discovery of CRISPR as a gene editing technology. Jim Allison, very important for immunotherapy research, a key player in that field, also Nobel Prize laureate. Also very interesting people from the more applied and industry. So there's people like Vishwa Dixit from Genentech.
He's from Kenya.
And he's from Kenya. So it's also important to have representation. People like Özlem Türeci from a chief medical officer at BioNTech. And also I thought it was really nice that some of the keynote lectures, as referring what you said, are research from people doing research in Africa, such as Glenda Gray, Henry Mwandumba, doing research in HIV, in tuberculosis. So I think that's also the list of people doing the keynotes as part of the preliminary program is very interesting for our listeners to know what's coming. And So I want to say, now that we're talking about involving, as you mentioned, researchers in this case from Africa, which is the hosting continent in a way. So I think that it's important to give them a spotlight, but also from low and middle income countries. So I just want to make sure that we let our listeners know that for those people that are applying for low and middle income countries, that there is a concerted effort to provide with scholarships to cover the cost of attending the conference. And I think this is very important for our listeners to know that that they can apply if this is their situation. So I know it's very close to your heart, Miriam. So maybe do you want to give a quick recap of what to expect when applying for these scholarships?
Yeah, well, this started because the statistics of attendance of trainees. I'm talking about trainees from low and middle income country in this type of meeting in prior big Immunology Congress was very low, right? So the challenge that we decided the challenge that we have is to really change, these numbers. So it used to, as I was mentioning earlier, like less than 1% of the trainees were coming from the low and middle income countries. It can be transformative to be exposed to this type of environment, right? So we want to change the optics of that, and we want to have 10% of the trainees attending our conference coming from low and middle income countries. The problem is that there is often not as much support for these type of conferences in low and middle income countries. So we have to fundraise for it so that we can cover the expenses of as many people, as many trainings as possible. And our goal is to cover 500 grants for our trainees from these countries. So we have progressed in our fundraising effort, but I'm hoping that we will continue to fundraise. So if people who are interested by this cause are listening to us, please reach out to us. We still have not reached the 500 number goal that we had set for ourselves, but definitely for trainees that want to apply, if you check the box low and middle income country, the fee that is associated with the abstract submission is waived. And our hope is that at least a certain number of those applicants will be fully subsidized to spend these 5 days with us at Cape Town. Deadline is May 2nd, so don't miss the deadline, please.
It's 2 months basically and change.
I wanted to add for listeners that want to follow the IUIS. So there's in social media, if you follow IUIS _online, then you will find all the information and be updated with all the data, the important dates, and the new stuff from the program. So the program is still rather preliminary. As I mentioned, some of the keynote talks are already settled, but there's a lot of space for people to bring their research and to present. Will there be poster? Will there be invited talks? What would be the so people can present for a poster presentation, also for maybe short talks? Is that also a possibility for trainees applying?
Of course, there will be, from those abstracts that are submitted, some will be selected for short presentation. We will have a lot of poster sessions, but the goal of the abstract is also to select for short talk, so which will provide visibility to this young. We have to this young scientists. We have another program called Rising Stars, which is focused on early career scientists, so faculty in the 5 first years of building their own laboratory, because this is where usually we struggle and a lot, and there will be a specific program to invite junior faculty also to give a talk. That's also another effort that we are building, and it's but is already advertised, and we already received application for that.
All right. So to kind of start wrapping our conversation. So the IUIS 18th International Congress of Immunology will take place this year in Cape Town, South Africa, between November 27 and December 2nd. So every trainee, every scientist in immunology is welcome to submit their abstracts before or up to May 2nd. And scholarship will be available for trainees from lower and middle income countries. And have you guys been to Cape Town already? Is there, for us, we're going to be there too. So also very excited about meeting everyone over there. Any highlights for those who already know the city?
There's a long tradition of wine making, and so there are wine regions, not right in Cape Town, but in outer lying areas.
All right, I'm liking this.
Just landing in the airport, it's a spectacular view of Table Mountain. That's just sort of right there and dominating the whole area, really. And then there are game parks, shark cages. You can go down to the Cape of Good Hope. You can drive down there, and it's a nice scenery and penguins and stuff. So that's a day trip out of Cape Town. So there's lots. It's beautiful. It's a beautiful country.
Cool. I want to go see African penguins. I've seen South American ones. I wonder if they're any different.
Just the accent. Yeah.
Just the accent.
My South African colleagues tell me that Cape Town is one of the most interesting cities. So they were pleased that we were going to Cape Town. It's going to be exciting. Please come and join us.
We will. And we're very much looking forward. And I think I really appreciate the as you say, the location. I think is great. And I hope to see people from all over the world. It's going to be super fun to attend, and a great opportunity for everyone to go and meet and network and start those contacts that will allow some breakthrough research to come to fruition. So it can only be a good thing. Before we finish, anything else you want to share with our listeners?
No, thank you for having us.
Yeah, thank you, and please, listeners, come. It's going to be terrific.
We'll be there, and we look forward to doing all the things. I mean, I've been excited ever since I learned about this a month or so ago.
IUIS2023.org is the website to find all the information, register, send your abstract, and stay up to date with the news of the program.
You are on top of that, Brenda. You've obviously been to that website recently.
I know. It's a beautiful website. Well,
thank you again.
Bye-bye.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at @ImmunoPodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.